Greetings everyone, we are from Group 6, Subgroup 1, and this is our video for Practical 2A, Spectrophotometry, Sugar Analysis using the 3.5 DNS method. Practical 2B, Determination of Sugar in Sample using the 3.5 DNS method. Practical 3, Physical Properties of different lipid and fatty acid. This is our group members. For practical 2A, the objective is introduce the student another example to the use of spectrophotometry for preparing glucose standard curve. These are the material used in the experiment. First, spectrophotometers, hot plate, glass beakers, cuvette, test tube and test tube rack, micropipette and tips, distilled waters, standard glucose solution, sodium potassium tartrate, and DNS reagent. Practicals 2A, experimental procedure. The first step, all reagents including distilled waters, glucose solution, and the DNS reagent are pipetted in duplicate into a series of dry clean, and the test tube are labeled 1 to 6. Next, the test tube are shaken well to ensure the reactant to be uniformly mixed for the reaction to proceed efficiently. Next, the test tube are boiled in a hot water bath for 5 to 7 minutes. This step will help to increase the rate of reaction between the DNS reagent and reducing sugar. Then, after the test tube being cooled, 1 ml of Rochelle salt are added. Next, the solution is transferred into the cuvette. Last step, the reading of exorbents are tested and recorded using spectrophotometry tools. The readings start from test tube 1 until test tube 6. Practicals 2B, the determination of sugar in sample using the 3.5 DNS method. The objective of this experiment to introduce the student and others examples to the use of spectrophotometry for preparing glucose standard curve. These are the material used in the experiment. Our experimental procedure for practical 2B is: the first step, add 1 ml DNS reagent into all test tubes containing sample as measured according to the tables. Next, the test tube are shaken well and boil the test tube for 5 to 7 minutes. Then, cool them down to the room temperatures. Then, add in 1 ml of Rochelle salt solution into the mixtures. Then, transfer the solution into the cuvette. And finally, the last step is record the readings. Practical 3. Physical properties of different lipids and fatty acids. The objective of this experiment is to investigate physical properties of lipid and fatty acid. These are the material used in the experiment: microcentrifuges tube, spatula, dropper bottles, distilled waters, toluenes, 3 selected lipid-based foods. Our experimental procedure for practical 3. First, prepare 2 sets of 3 microcentrifuges tubes and add 5 drops or 3 to 5 granules of each lipid containing product you brought.